So this is all about questions, and it's a little bit about history, and I do a lot of talk about history. And the history I'm going to share with you today is really about the creation of Celator, which is a company that you heard about yesterday that was bought by Jazz Pharmaceuticals for $1.5 billion. And that took a long time, and it was an interesting experience. Many of the people in the room have joined that experience and lived that experience, so it will get their perspective on it as well. And I'm stupid enough to actually go forward with the initiation of another company called Cuprous Pharmaceuticals. And Cuprous is about 4 years old. I'd say it's only 3 years old. And a lot of the issues that I encountered in the context of Celator are now being reflected in how we're changing the operation of Cuprous. So where does this all start? And I sort of hate to say this, especially after what Peter just said. It started with grants. And it basically was a grant on a multifunctional liposome delivery systems. And I actually do this almost as a joke. It was not necessarily liposomes, because in the mid-90s, liposomes were a bad name. So somewhere along the line, I'm not pointing any fingers at anybody, but somebody came up with this lipid nanoparticle. So basically, my rejected CAHR grant, I did a search and replace of liposome with LNP, and the grant was funded. It's like, wow. So I'm back to liposomes today, but there was a reason for that change. That multifunctional liposome formulation was really about developing drug combinations. And you always have to reflect on the history of how you got to what it is that you were pursuing, even academically. And that academic vision that was captured in that grant, developing drug combinations, arose out of work which was done with Peter in the liposome company and the development of a drug product called liposomal vincristine. It's now Marquibo, and Marquibo is being marketed in the States. I was responsible for the development of Marquibo in the Canadian Liposome Company, which was owned by the Liposome Company. Liposome Company had a very strong cancer scientific advisory board. We were invited there at one stage, I'm going to say 1990, maybe 1989, so some time ago. And I had to give the presentation on liposomal vincristine, and Peter may have a different version of the story, but my version is, I opened my mouth, I said, I'm going to tell you today about liposomal vincristine, and somebody said, why are you doing that? And it was like one of the who's who in cancer, a guy named Dr. Frey, Emilio Frey, Tom Frey. He's the guy who invented drug combinations. He's the guy that actually took childhood leukemia from a stage where you were having 70% of those kids dying to a point where they had 90% of those kids living with the disease. So this guy did amazing things, and it was a slow process. It was taking drugs in combination. He came back and said, in the context of liposomal vincristine, why would you ever develop vincristine as a liposomal drug? Nobody uses vincristine in isolation. Vincristine is always used in combination. So that sort of set the stage for me thinking, I should really be thinking about drug combinations. It helped that we were let go from the Liposome Company. So that was our first firing event, and that's an interesting story which we can share later. But again, I'm going to give credit to Peter. Peter knew that we were going to get fired. He told us all that we were going to get fired. So we were well prepared for the fact that we were going to get let go. There were still people that were upset about that whole thing. So it was interesting. So the story of Marquibo, I've already got that. You can see I've got notes that I have to go through. So the goal that we embarked upon is, how do we go about developing a drug combination product for use in the clinic? And that really was the inception of Celator. It was a very simple concept of, can we take two drugs and use them in a manner that would come up with a more therapeutically active agent? And I would say at that stage, the founders of the company, which is Paul Tardy, Ann Wallace, Lawrence Mayer, and myself, struggled a lot. We had a lot of academic credibility, but we had zero business credibility. But we felt we had a fair bit of business credibility because we were associated with the Liposome Company and the Canadian Liposome Company and INEX and Lipex Biomembranes and Northern Lipids. We had, we know what we're doing. We can do this. Well, it turned out we really didn't know what we were doing. And on top of that, people really came back to us and said, you can't do drug combinations. And I did my academic thing. I said, cancer's always treated with multiple drugs. That's the way cancer is treated. So of course you can do drug combinations. Then I said, look at the AIDS community. Look at the changes in the population treatment of AIDS patients, going from multiple drugs to combination products, which are really making a difference in the life of those patients. And these guys kept on saying, you can't do this. And I'm going to talk to clinicians. And the clinicians would come to me and say, you can't do that. I was like, really? So it was just, no, you can't do that. All the way around. So criticism, you have to be able to live with the criticism that you get both academically and from an industrial perspective. So when Kelley rejects Cupris, it hurts. But I understand. I'm going to get in a little bit about the history of Celator again. So we had this idea. We actually had an angel investor who was technology naive, just a nice guy who really liked Lawrence and myself. And we were going out and asking friends and family for $25,000 each. And he came back and said, what would you do if I gave you $1 million? When we go, we could use $1 million. So we took the $1 million and we spun out a company. We went from a virtual company within BC Cancer to a company outside of BC Cancer. We expanded our workforce and everything. We were great. We were just going a mile a minute. We had IRAP funding. We had CIHR funding. Everything was wonderful. But we knew in order to expand, we needed to get venture capital money. So we put together business plans. We had marketing advice. We did all this lovely stuff. We went to the local VCs, and I'm not going to name names, and I'm not going to try to say evil things about these VCs. But I do reflect on one conversation that we had with VCs, because we actually had a lot of credibility. And the VCs actually organized a consortium. So there was like 4 of these VCs in one room, and we were presenting, and we had a well thought out, well-constructed business plan asking for $30 million. We knew we needed $30 million to do what we said we were going to do. And one guy, and I'll mention his name. What's his name? The guy, the finance guy at INEX. I want to say, Brad, I forget his name. Anyway, he's working for Genome Canada now. Anyway, I won't get into the names. But he basically, after we do this lovely presentation and we're confident, we know what we're doing, Pat Brady, yeah. So Pat goes, well, we can't give you $30 million. What would you do with $10 million? Or would you accept $10 million? And I'm not going to say who said it, but one of us said, oh, that'd be great. And we were escorted out of the room, and that was it. That was the end of the company, basically. We knew that we had lost in a single statement 100% of our credibility, because we developed this detailed plan, which was really precise, and I'd even say today was precise, that we would need $30 million to do what we wanted to do. And they came back with 10, and we should have come back with, we have to go back and try again to see what we could do for $10 million. But it's not going to be this plan. But instead we said, we'll take that. So the message is, think carefully about how much you're asking for and understand what that money is going to be used for. In order to overcome that barrier, we ended up having to bring in somebody from the outside. And we ended up hiring a person from the US, I'm not going to mention his name either, who became the CEO. And he did some great things. So at one level, he brought credibility to the company. So all of a sudden we had a different face meeting with the venture capital people, so that allowed us to get money. He got us organized and he focused us, which I think was really, really pretty important. When you talk about, we're doing drug combination, it was like, there were so many things we could do. But did any of that actually make any business sense? And he struck a scientific advisory board. You heard that yesterday. That advisory board came back and said, if we're going to do this, if you want to do drug combination products, focus on drugs that are already approved by the FDA for use in combination. So we ended up focusing, and that was great. So we got money. There's two things here in this note. So we got money. We got $10 million. There's a long story behind that $10 million that we got to get us going. And then he fired us. Fired me, I should say. I won't say us. And I won't say he fired me. I quit. And I actually view it as constructive dismissal because I was unable to do my job effectively working in Cellator. And I was bitter at that stage, and I would say it took years to get over that bitterness. I'm over that bitterness now in the context of understanding that my role within that company probably needed to come to an end. And that's one of the messages I would come to you as you're starting a company. You have a role in the development of that company, but that role is going to change as you move forward. You need different people, different expertise. You need different skill sets. The other thing about that funding event was I started off before getting funding owning 46% of that company, all by my little self. It was awesome. I walked out of that funding deal and I owned 0.8% of the company. So you can see what sort of dilution might actually come with this. And related to that dilution was the need. When we went to the venture capital group, and I don't know if this is uniform across all venture capital group, maybe Ewan can say something about that. They knew everything about us. They did deep due diligence, and they knew exactly how much money we had left. And they took us to 2 weeks to having no money. And they put forward, after that point, money on the table. So we were really obligated to take that money. I don't think that was fair, and I think in hindsight we should have taken a different approach, but that's just becoming aware of what's going on. Throughout all this, I had multiple hats. So as Cellator developed, I retained my position at BC Cancer. I justified that the same way I'm justifying it today. We have grad students, we use space at BC Cancer. There's a reason for maintaining that association with cancer. The problem that I ran into is when I left Cellator, I was bitter. I actually went and talked to a lawyer. I thought I was constructively dismissed. And the lawyer came back and said, well, you might have been. You could take this to court if you want to, but frankly, you have a job and you're well paid. You're not going to get any money from this. Nobody's going to listen to you. So it's sort of like, huh, okay. Back to my academic lab, be bitter for a while. Start Arena 4C, and I didn't even mention that as a story here because that's an interesting story all in its own right. It's actually one of the ones where if you do not move fast enough, you are going to be overtaken by somebody else. So at the time when we developed Arena 4C, which is an irinotecan in a liposome, it was probably one of the better drugs that we've ever developed since we started developing drugs using liposome technology. And it was at a time when there was another company that was also developing a liposomal irinotecan. In that context, we, I think, were ahead of that company. Within a short period of time, 2 years, we were behind that company. They were moving into clinical trials. And I was still going, oh, I wonder if you should start a company. I don't know what I was, we were wobbling. And by the time I said, oh, we're going to start a company, it was too late. And we also ended up having loss of control of the technology. So while this was all going on, BC Cancer was out trying to out-license this technology. That technology was out-licensed to another company in the States. Huge celebration event. Everybody was happy. This was the greatest thing since sliced bread. We thought we were successful. That company sat on that technology for 2 years and then returned it back to BC Cancer because they didn't know what to do with it. They didn't know how to do with it, anything with it. And they believed that all the work that we had been doing on it to date, and that included manufacturing and GLP toxicology studies, had to be repeated. That was their advice. So it was a good event, but because we lost control of the technology and it took a long time, it died. So where am I here? So the messages that I say that we've learned, you have to accept the fact that you need to change. One, you need to embrace new ideas and move forward with them. You have to accept the fact that your role in a company is going to change as it grows, and that you may have a specific role, or you are going to have to re-educate yourself and to a different role. The other message I had here, and I think it goes back in time to many of the companies I was associated with, was the idea that you need to do this in partnership. I work in the area of oncology, and to develop an anti-cancer drug is stupidly expensive. I won't say it's the 1.5 billion that the pharmaceutical companies say, but there's a group in Vancouver Island that did a pharmacoeconomic study, and it really is $100 to $150 million to develop an anti-cancer drug. It's a lot of money. So I think trying to do this yourself is a trick. You need to do this with partnership. And the one thing I did learn, and that certainly was a learning event in the context of Celator, is an inability to develop a partnership. And so Cuprous is now formed. We have new technology that came out of VC Cancer, which is being developed for a number of different applications. Our focus right now is on the immuno-oncology area. And our goal, our single goal, is to develop partnership. And what we have learned is that we need help doing this. So we actually ended up getting, and I have to put this in the right way, we were accepted into a program called E at UBC. It's Entrepreneurship at UBC. I know that all the universities across Canada have similar programs. And this was a program which was basically a class. You go into phase 1, phase 2, phase 3. And in order to get to phase 2, you had to pass phase 1. In order to get to phase 3, you had to pass phase 3. And if you passed phase 3, you ended up into something called Hatch. Luckily, Cuprous has actually now made it into Hatch. But at each one of these stages, you ended up having to do homework. You were given exercises. You had to do reports. In Hatch, we have to, I won't say fake, but we have to do board meetings. We have to present board books to this panel. They're not our board, but they're, sort of treating us as if they were our board. So it is a learning event, and they are embracing this idea of how do you develop a partnership, and they're teaching us now how we approach that partnership. And the only message I'm going to give you here before I stop talking, how much time do I have? Probably lots of time, but 3 minutes. Okay, I have great time to stop. So one of the messages I got through E at UBC and Hatch is identify the bleeding neck wound of a partner. And I never heard of that phrase before, this bleeding neck wound. And it really was a description of what does that partner need? If you go to a partner, which is what I have done in the past, saying, I am great, my technology is great, I know everything about this area, and what you're doing, it's just a waste of time, and you need to talk to me because I'm going to solve that problem. That is the wrong approach. But if you go in there and you say, you have a problem, or you can't even do it that way. You can't say you have a problem. You have to go in there and understand what you think their problem is, and then you have to go talk to them and say, do you have a problem? And then you need to have a question about, is there a way that our technology can help solve that problem? So unless you can identify what that problem is, unless you can recognize, the company itself recognizes that problem, you're not going to be able to develop a partnership. Now, has Cuprous developed a partnership? We've got one. So I guess that's okay. But we certainly have a goal of having 3 at the end of next year. So I think it's going to be a challenge. We have over 400 companies that have been identified that we want to talk to, and we'll see where this goes. And hopefully we've learned a little bit along the way. Open for questions. Well, I'll add something about that initial investor. He actually came in and gave us another $1 million. And I knew nothing about what was going on in the financing of this company. I knew what was going on. I knew we were getting money. I knew we could continue to do our work. But I didn't know anything about something called like AntiDilution Clause. Which, how many people here know what an AntiDilution Clause is? Yeah, so some people here know what an AntiDilution Clause is. So this early investor was clever enough to put an AntiDilution Clause on his investment. So as I went from owning 48% of the company to less than 0.8% of the company, he went from owning essentially 4% of that company, and that 4% was retained until the company was sold for $1.5 billion. So it took him a long time, I'm going to say 16 years, to realize that investment, but he actually made a lot of money. So he was clever, and I think I agree with you. I think I would have used, in my knowledge now, that $1 million in a different way, but I think Celator probably wouldn't have existed without that $1 million. The CEO that we brought on to help us from the US, and Peter knows the individual, and I have really harsh words to say about him, Celator would never have succeeded had it not been for that individual. So sometimes you have to bring in the right people to do the job that needs to be done. So, yeah.
Other questions or comments? Yeah, Gilbert.
How did you choose a scientific advisory board since you knew everything about the science?
This is going to sound really weird, and I'm sure Peter will be able to realize how this happened. So the idea of doing drug combinations came to me out of a scientific advisory board meeting for the liposome company. Celator was created, and within 3 months of the CEO joining us, we had a scientific advisory board, Cancer Scientific Advisory Board, which was essentially identical to the same scientific advisory board that liposome company had. And it was those people that were enthusiastic about drug combinations, and they really were the who's who in oncology in the US, so tremendous amount of credibility. And we expanded using that information for Celator. When we think about Cupris, we don't have a scientific advisory board, but that's because we have lots of advisors. So we've reached out, I'm going to say, to more than 50 well-known scientists. Of those 50, we're going to have to probably narrow that down to 8 scientific advisors. And then the board is a completely different story. So the board needs to be settled, and those are going to actually impact how the company operates and grows going forward. The board of directors, yeah.
Can you tell us a bit about that? How do you, so it sounds a little bit as if you lean on connections to get people into these boards. Can you tell us, how do you do that?
Well, I think I'm going to...
You imported the entire scientific advisory board. What about the
I'm actually going to leave that for other people, because I'm going to say that, I have said I wear multiple hats, and I do, but I also know where my limitations are, and people sort of laugh at me at that. Well, and one of the things my limitation is business. I don't like it. I'm not here because of the business. I get laughed at because I'm not here because I want to make money. My credibility comes, and I'm going to say this, and actually, again, I'll give Peter credit. It comes from this thought of I'm old. I'm going to be retiring soon. But I believe strongly that I have one more drug in me. If we have one more drug that we can actually get approved, and that is a long process. So that is a process of 10 to 15 years to get something to the point that you're getting approved. So I believe strongly that we can come up with something that's going to be approvable, and that's what's going to be going on. But it's not going to be me. It's going to be a whole bunch of other people. Paul Tardy, people met Paul Tardy yesterday. Again, I would say Celator would not have been successful if it had not been for Paul Tardy and the team that he had working, trying to sort out the formulation that allowed 2 drugs to come out at the exact same rate. It was a very, very challenging thing. So you've heard me say, in just in the last 2 minutes, 3 people, the initial angel investor, the CEO that we brought in, Paul Tardy for doing this rigorous work in the characterization of the carrier, that was what was responsible, plus the other people, plus the clinical trial person that was in charge of developing the clinical technology. It's a huge effort to do this, so you have to embrace that, and you can't come in there and say you know everything, because you don't. And I think I'm good at the science, I think I'm good at the IP, but I'm not good at the business. I'm lousy at the finances. Ask my kids.
All right, we have one more quick question.
How are you identifying those potential partner organizations, and how are you
So that's Hatch, and they're helping us, and I'm gonna get the names wrong. You can tell I'm not into business. Is Ada here? She's not here. She's going to a wedding on Galliano. There's spreadsheets out there. There's databases out there of all these companies, and they are deep databases. Kaylee, do you know the name of these guys? UBC has access to them. So, I mean, it really, it says, when was it founded? Who was the angel and founder? What are they doing? Where are they in the creation of, Pitch book, yeah. So it's really, really, your university should have a copy of this, and it's really quite restrictive in terms of its access, but the detailed information there is very useful, and I know I should shut up, and I'll go on to one story. So this idea of going into a partnership, we had a drug product that we wanted to develop in a partnership with a company. Kent gave a presentation on this particular drug at this meeting recently, and we identified the company using Hatch and the support of Hatch, identified how we should approach that company, and I think we did a really good job introducing ourselves, trying to set up a time to meet with them. We actually were going to go to San Diego to meet at ASH with them, and then they canceled the meeting. And it's sort of like, oh, what the heck happened there? And it turns out to be actually quite simple, that company was owned, and we knew this at the time, it was owned by a parent company in Japan. And did we do any homework? Did we understand what the Japanese company was? Did we know anything about that? And we didn't. And had we done that, we would have made a much better approach. So right now, our list, the pitch book list, I think, is 8,000 companies. That's huge. And we've now narrowed it down using a bunch of different strategies to about 400 companies, which is huge. And when I talk about in-depth analysis, is who are the scientists that created the company? What is the technology that they're developing? Where are they at? Who's funding them? What are their problems? Can we address their problems? It's a big job.
Well, thank you. It's wonderful to be here. And what I'm going to tell you about today is actually a set of 3 short stories. I promise they'll be very short. And one of the themes here is that, I've always thought that the term serial entrepreneur is kind of redundant, because most people that will go out and are crazy enough to start a company will start another one and another one and another one, right? And are a little bit addicted to that process of getting things going and really trying to push to get things out there. So I'll tell you these 3 stories about 3 companies, but I wanted to start with this. So students and other people ask me all the time, Shana, how do you come up with ideas? Like the ideas that you pursue in the lab, the ideas for the startups. And I kind of laugh, because it's like the idea generation to me over time has become the easy part of it. Pretty soon you have way too many ideas, right? My students will definitely tell you this about me. Shana has way too many ideas, and we really wish she could, focus a little bit. And I think this is an important thing for trainees to know, that once you get going with the creative process and coming up with ideas for research and companies and the whole thing, it will just keep going and going and going. Your ideas will get better and better. And then I think the other important thing to realize about startups is that you will not have it figured out on day one. And you will be, building the bridge over the cliff while you're driving, and it will be very exciting because you'll be figuring it out on the fly. And we all get better at doing this as we have more and more experiences, and we try not to make the same mistakes over and over again. But it is definitely a learning process. So here are the 3 companies that I'm going to tell you about. And you'll see the connections and how really one was kind of the leaping off point for the next one. And I hope this list will continue to grow. I think we still have lots of good ideas to pursue. These are all diagnostic companies. So these are all ventures where we set out to try to provide new diagnostic tools to address really urgent needs in the diagnosis of infectious disease, cancer, deal with adverse drug reactions, all of the areas in which we need new solutions to provide patients with better care. And really the big push here is to try, we talked a little bit about this yesterday, getting diagnostic testing out of the lab, right? Can we finally push past this paradigm where we take samples from people, we ship them off to a lab, we batch them, we wait a few days for them to be processed, the results go back to the physician. For some indications, maybe the physician has already gone ahead and treated the patient based on some guesswork, or maybe the patient's about to find out that they have something that they really don't want to know they have, and so they don't call back when the physician calls them. It's a very ineffective system that we currently have in place. So can we bring diagnostic testing closer to patients, allow physicians to do their own testing, give them a source of revenue, but also give them a source of really efficient ways to gather molecular level information about their patients, what's going on, and then to be able to choose therapy based on real information. And so that's really something that I've worked a lot on, and you'll see that really being some of the rationale for each of these companies. So the first story starts at this place. This is Caltech. This is where I did my PhD. And Caltech, if you've never been there, it's like the quintessential ivory tower, right? Like people are just walking around thinking deep thoughts, most of the time just thinking about very basic science. You know, there is a lot of entrepreneurial activity at Caltech, but there's also really a tendency to focus on the very basic aspects of scientific discovery. And when I was there, I was working on a project that was very basic in its nature. We were basically looking at DNA molecules and trying to understand whether DNA as a material acts more like an insulator or a conductor. We didn't really have any reason for wanting to ask that question. We just really wanted to ask it and answer it, right? This is how basic science gets done. And we learned a lot about that. We used ultra-fast femtosecond spectroscopy to look at that. We used electrochemistry, all kinds of really interesting scientific methods. But we eventually stumbled on something pretty interesting, and that was that if we did put DNA on the surface of an electrode and then measured electrical currents traveling through DNA molecules, that if the bases were mismatched, that the electrical current that we could monitor would get shut off. And so we had this way of detecting basically a single base change in a DNA sequence. And this was in the late 90s. We were not yet sequencing DNA the way that we are now. And so being able to look at this kind of a change in DNA was a pretty big deal. We didn't really have any ideas about exactly what to do with that. But eventually, you know, we decided to kind of take the next steps. We filed a patent, and this was myself and my graduate advisor and a couple other people. And then, I mean, even though Caltech is very much a place where people are focused on basic science, even at this time, they had an amazing tech transfer office. And so my advisor and I kind of wandered down to the tech transfer office one day. We explained, you know, this is our technology. We're not really sure what we could do with it, but we think it's pretty cool. And I don't know, maybe we should think about starting a company or talk to somebody about it. And I still remember this so vividly. You know, our tech transfer director, he had a phone on his desk, and he had Kleiner Perkins on speed dial, you know, on 1, and Sequoia Capital on 2. And, you know, this is at a time when anybody still used speed dial, and he made calls. We had, 10 meetings 2 days later, and we had a term sheet for $5 million then, 2 weeks after that. I mean, in those days, venture capital, there was kind of a bubble that was building, and it was pretty easy to access it from a place like Caltech. So we eventually did spin this company out, Genome Sciences, and we did this. I finished my PhD. I went off to Boston to start my first academic position, but I flew back and forth, East Coast, West Coast, helping to get the company started. And so eventually we ended up developing these electrode arrays that allowed you to test for specific DNA mutations. And we thought that the best place to deploy this would be on looking at different types of genetic disease. And we had prototypes, and we had some assays that worked really well, and we had some clinical studies that were done at places like the Cleveland Clinic. And we really thought that this was going to be the first set of products that would get launched by Genome Sciences. But it turned out that our management team had a slightly different idea, because the genetic mutation testing idea sounded interesting, but at this time, bugs like methicillin-resistant staph were the headlines. Were all over the newspapers. People were really starting to freak out about antibiotic-resistant infections, again, all over the New York Times, and the trajectory in terms of the rising rates of antibiotic-resistant bacteria was just going up and up and up and up. So our management team basically said to the scientific team, guys, we got to change gears. Like genetic testing, all fine and good, lots of merit to doing that, but we got to get on this right now. So we ended up actually buying a Canadian company, and got some access to some IP around the genes that allow you to say that a methicillin-resistant staph is methicillin-resistant. We were going to take those genes and the ability to develop assays using them and merge them with our electrochemical platform. And eventually, we're all very excited about this. But we got a call one day from one of the bigger players in the diagnostics space, Becton Dickinson, offering us quite a bit of money to just sell. And it was, people were torn. We felt like we had a lot to do. But at the end of the day, you just don't walk away from an offer like this. So we did sell to Becton Dickinson. And the wonderful thing was that they took a lot of the elements of what we had done, and they launched an entire product line based on infectious pathogens that are problematic within the healthcare system, especially in hospitals for immunocompromised people. And they really built a very good business based off of this approach. The unfortunate thing was that they took the electrochemical platform and put it on a shelf somewhere and never did anything with it. And so the money was in the bank. It didn't really matter. But just as a scientist, you have those pangs like, oh, I wish that had kept going. So in my own labs, we did then try to kind of resurrect the idea, but really push the performance of an electrochemical detection approach. And so we developed the sensors that are shown here. These are three-dimensional sensors that are made out of nanostructured metal. They have lots of neat materials chemistry that go along with them. And so we started to think about taking those sensors out into a point-of-care testing system that could be used, again, for this kind of near-patient diagnostic type of approach that I mentioned a few minutes ago. And I'm trying to see if the filing date's on there. Yeah, so 2010, we got this technology off the ground really in 2008. Started making calls about, want to start a company, maybe we could do this, do that. There was a lot of enthusiasm about what we were trying to do. But then I think it was like September of 2008. And I think we were like getting very close, like cruising towards a term sheet. And then Lehman Brothers went down. And then nobody called us back. It was like, goodbye. And everybody in this room remembers this time. I mean, wallets just snapped shut, right? You could just hear that across the world. And so we had to go into a holding pattern for a couple of years. But we did that, and we waited till the economy kind of rebounded and then got restarted with the fundraising process. I counted one time how many meetings I had to have until I finally got a yes in terms of finding a Series A investor. It was 90-something meetings with different organizations, and maybe it was 94. 93 of them said no. Only one of them said yes. And I think Marcel mentioned this, like, the thick skin that you have to develop to be in this it's one of your most important tools, because you can't get discouraged, because people will just say no, 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 no. You realize that most of people, like, no is just a default position. It is what they want to say, and you have to convince them somehow to say yes. And it is a really difficult process and really very challenging. We finally got this company off the ground. It was called Exogenic. We built out a facility at 55 York Street. If you know Toronto, this is right next to the Royal York Hotel on York Street. And this was a slightly funny location to do this, but it was the only built-out lab space we could find. And so we ended up building a test, a combined STD test for chlamydia and gonorrhea. It's a big market, over $1 billion. Clinicians really like it. From a regulatory standpoint, it's a good one. There's existing reimbursement. So in focusing on this as an indication, it really checked all the boxes that you need to, as best you can, when you're starting to get something new off the ground. We didn't think there was market risk. We didn't think there was regulatory risk. And we didn't think there was reimbursement risk. There were other kinds of risk, which I'll get to in a moment. But we had checked some of the big boxes. And so we got going. We raised some money first with Mars Innovation, and then came the Series A. We developed a completely integrated testing system, first as an alpha prototype, but it worked. It did testing. We moved on to a beta. We put a lot of work into the industrial design of this system. We really wanted it to have kind of a consumer electronics look and feel so that it was really easy and intuitive to use. And then we started thinking about, okay, so what comes next? We got to take this system. We got to take this STD test. We got to get it out there. We got to do a big clinical trial, because this is actually a screening test. We've got to look at thousands and thousands of patients. How are we going to do that? And we realized that there was a whole manufacturing piece that had to go with this. And we had to learn to make hundreds, thousands, tens of thousands of our testing cartridge. And so luckily, the investors that we had were kind of willing to come along on that ride with us. And this turned out to be a huge undertaking. We had to build a clean room. We had to build, the entire manufacturing line to do pilot manufacturing. And because of our location, what this entailed was actually shutting down York Street for a Saturday and craning all of the walls of the clean room and all of the equipment, $5 million worth of equipment, into this facility that we had, which was on the 16th floor of an office building. I mean, it was kind of a nightmare. And I remember standing on York Street this day with my kids who were really little at the time and watching, these huge boxes getting hoisted up. And I just felt like my whole life, was like dangling from that crane. And this was really, this was quite an experience watching this all get put together. But we did it. We built the facility. We were able to make this system. There was a lot of drama that went on that I won't tell you about during this. We hired an external management team. I really personally had raised all the money for the company, $40-something million. We brought in management. I kind of then watched management spend the money in ways that I wasn't always very happy with. And that's one of the big challenges when you're an entrepreneur, is kind of trying to let go, even when you feel like maybe things are not being done completely the way that you would, but you have to just, let it go. And so we did have this system that came together beautifully, clinical grade, built with FDA compliant procedures and everything. And we took it out. We did a beta study in a local STD clinic, showed that the system really worked. It had performance that was equivalent to the test that would be done in a lab. We could provide, really real-time information, 20-minute turnaround time. And so this was all really wonderful, but we also knew that we had an even bigger challenge in front of us. So we started to look at the dollars that it was going to take to continue scaling manufacturing. And if you look at the price point for this test, this is a $75 test. The healthcare system will not pay more for you to figure out whether you are infected with chlamydia or gonorrhea. $75. And so when we looked at what we were paying per cartridge at the time, it was about $200 to manufacture one unit. So we were going to be, taping a $100 bill to every cartridge that we sold, right? This is the problem with scale up. And it was going to take huge infusions of money to get us past this. And we shopped this around to a lot of venture investors who just, were like, no, thank you. You're going to scale up. You're going to do all this. You're going to have a product with a gross margin of like $30. You're going to sell 1 million of these. And, there is a nice hockey stick, but we don't like the y-axis, on your revenue projections. And we were, again, like $43 million in and just looking at this square in the face. So this was a really difficult time, to put it mildly. But eventually, so financially, this was not a huge success for the investors, but we were able to find a buyer for the technology to allow this to keep going. And so we teamed up with General Atomics, which is a U.S. defense contractor, best known for developing the Predator drones used for surveillance throughout the Middle East and all over the world. General Atomics wants to start moving into kind of more peaceful types of activities. And they're now doing big contracts to outfit military clinics with point-of-care testing instrumentation. And so they were able to take the technology, and you can see how this economic issue, it goes away. Right now, if they're selling to the military, not into mainstream clinical medicine, they can charge $300 a test, $500 a test, right? That barrier kind of goes away. And so this was absolutely the right way to scale this technology. Again, not what we envisioned, but I'm happy that at least the technology is in somebody's hands and is moving on and will make an impact. And then the intention is to, once it is scaled, spin it back out and then factor it into mainstream clinical medicine. So I'm probably pretty much out of time. I'll get to my last story, which I can absolutely do in 3 minutes. So then this comes to the most recent company. As we were developing the Exogenic technology and looking at all the things that it could do, we also realized that there were some things that it couldn't do. And in looking at rare cells, looking at liquid biopsy applications where there's not only a really significant sensitivity requirement, but also specificity requirement, also a lot of heterogeneity that you have to deal with just because of the nature of cancer. This gave us some ideas for where to go next in terms of new technology. And so over the last few years, we've been working away at developing new techniques to look at circulating tumor cells and really profile them and collect molecular level information from them for therapeutic decision making. And so this is something that we now have some IP on. and are rolling this out into a company that's really going to be focused on therapeutic decision making and trying to collect information through liquid biopsy that allows immunotherapies to be used more effectively, many of the molecularly targeted therapies where you need real-time information about what's going on with a patient's cancer to know what drug is the right one to administer. And so I'm very excited about this. We have about $1 million worth of seed funding coming from Facet, which is an arm of the Ontario Institute for Cancer Research. And we're also very focused on partnerships. I think Marcel really covered that nicely, that as a small company, what I think one of the best things you can do is look out there in the space and see who you can team up with in order to deliver new types of tests and therapeutics as well. So this is where we are now with a new venture going forward. And so I thought I would just end with some things that I think are really essential elements. Number one is solid science and IP. I have learned over time, and I didn't really talk about this, but there was a lot of, in both of the startups, Genome Sciences and Exogenic, there was a lot of scrambling around, like the technology works, but kind of, like it worked in the lab, but now we've got to make it work every time. And honestly, you don't really want to do that in a startup. You want to do that in your research lab where you don't have venture capitalists looking over your shoulder. You don't have, a staff of 20 people that have left, jobs to come join your team. And, the science has to be completely worked out, and you have to know that it is really going to roll into a product. It's really essential to have a well-vetted product concept. We get ideas about how we can turn technologies into products, and honestly, even my own ideas, they were a little half-baked, and I just kind of come up with this idea, read a little bit of literature, and think I know what the product's going to look like. You have to get out there. You have to talk to people. You have to really try to punch holes in the product concept and make sure that it's real. A great team is absolutely essential. I have people that I've worked with in startups that have been amazing, so committed and really giving 1,000% to make something successful. I've worked with other people that I am legally bound not to say anything about, that they almost sunk all of these things that I'm telling you about. So it's just so critical to have that great team. And then lessons learned, building products takes a really long time. I'm not a very patient person, and I find this excruciating. It doesn't take a year to build a product. It doesn't take 2 years to build a product. In medical, highly regulated device, it takes 5 years, if you're lucky. So you have to have that timeline. The other thing that I've learned over time is that the best way you can go is to under-promise and over-deliver. I get a little bit too excited. I get very excited. I want to over-promise. This is going to be great. We're going to change the world. It's going to start spinning the other way on its axis. I just, the enthusiasm gets the best of me. Investors really don't like it when, you said you're going to have something done in a year, but then you tell them it's actually going to take 5. It's just not a good dynamic to establish. So, finding a way to combine that enthusiasm with the ability to be a little bit conservative, and even though you don't want to, really take a good look at the worst case scenario and make sure that that's really in your plan, that could happen. And then I love this chart. And so with cellular analytics, we actually went through the NIH I-Core program, which is a great program for vetting product concepts and really getting things right at the very beginning of a commercialization exercise. They're very focused on Steve Blank's philosophy in terms of commercialization, entrepreneurship. And this chart comes from that exercise. And I love this, that the number one reason that startups fail is because they build products that nobody wants. And it's not, they're not scientifically interesting or exciting or whatever. It's just that there's no market, right? And I get so obsessed with the technology side of it that you have to recognize that you're building something that people are going to have to buy it. And so you have to look all the way to the end of the process very early on, actually. So with that, that's what I wanted to tell you. Thank you very much.
I mean, one of the issues that I think everybody who does any company starting hits is when you hire somebody in as maybe a CEO or whatever, and I have to admit my failure rate is rather high in that regard. What's your experience?
Same. It's very hard. I mean, you have to be crazy to take a job as a CEO at an early stage company, right? I mean, it's not a safe bet. It's a difficult position. And you're coming in, there's already a bit of a dynamic there, so it's hard. So, I mean, finding that right person, at Genome Sciences, we did have an amazing CEO. And the reason that we did that, did the acquisition with BD is because of that CEO. And we just got incredibly lucky. Having top tier investors can really help you with that, because that can kind of help people overcome the uncertainty around an early stage startup. But I think this is one of our biggest challenges in Canada. We're not in Silicon Valley, we're not in Boston, we don't have a critical mass of these kinds of people around. And so we had a situation where we had somebody commuting from San Francisco. Terrible idea, absolutely terrible idea. I would never do that again. It just can't, startups are all about intensity and the team and pulling together. And when you have somebody who kind of pops in, every other 2 weeks after they've just got it doesn't work. So I think it's a big challenge.
The School of Hard Knocks, I thought I'd take this, give you 3 examples, such as Shana did. I'm going to go through the good, the bad, and the ugly. Some of it's, quite ugly. These are 3 companies that I've been involved with, Lipex Biomembranes, Acuitas Therapeutics, and Arbutus Pharmaceuticals. All of which are still in some form or another, still in existence, but certainly some interesting histories involved there. Okay, I've co-founded way too many companies. I mean, this is. I don't know quite how this has happened, but we now have, I don't know, it must be on the order of 8 or 9 or whatever. I should say none of them have failed. So this is one thing that I think, just to give people some reassurance. If you really do have some solid science and some solid people working with you, you're going to find a way through this story about 1 in 10 failing, or only 1 in 10 succeeding. Well, I haven't experienced that, and I think if you're coming from a reasonable background, reasonable intellectual property, you have some good people working with you, that the odds are much better than that. So don't let those things scare you too much. But anyway, so this is starting in my lab back in about 1978. We started the Canadian Liposome Company. We started a couple of companies. We started these more or less by accident, and I'm just going to give you at least the initial one, and I'll give you some indication of how that led then to subsequent events, shall we say. I should say we started also a personalized medicine initiative. There's a couple of companies that have come out of that as well in more recent times. Okay, so let's just go through the good, which is a company we formed. This is the very first company that I was involved with in 1985. Lipex Biomembranes. We'd invented a device for making liposomes in the lab, and so one day I was giving a talk on this. I was in New York or something, and so somebody in the audience, and I talked about doing this extrusion technique, and somebody in the audience says, that looks good, I want one of those. And so I gave him, this was before internet and anything else, so I gave him the phone number of the machinist that we'd had make this thing for us, and we're in Port Moody, and 2 months later I got this very irate phone call saying, this thing is a piece of crap, it doesn't work, because this machinist had made it and sent it to him, and of course the machinist had no idea what it was meant for, so there was no quality, no testing to see whether it would actually do the job or anything like that. So we thought, okay, well, maybe we should put up a company and we'll do that and we'll send it out. So that was the start of Lipex Biomembranes. Now, we knew a few things about companies. So this is inventing the device, formed the Lipex to market the extruder. But the things we knew about companies were pretty basic. We knew we had to have a mission statement. So, okay, well, we got together. We thought, what mission statement do we want? Well, we came up with free beer every Friday and parties whenever possible. this is the best mission statement I have ever come up with. And we actually did it. We had free beer in the lab every Friday for 25 years until this company got sold at some point, and I'll get into that, and some monumental parties. So this is the extruder. The first version was made of plastic, which made it rather dangerous. Marcel can remember that because we were putting a few hundred psi pressure on it. But we wanted to be sure that the material we put in was actually going down and then coming out. Anyway, it's extruding through a polycarbonate filter, 100 nanometer pore size, and you can get the particles out the other end. Moving to a stainless steel version of this, which is what we've ended up selling. Well, this wasn't quite as satisfactory because you couldn't see the material going down. You could see it coming out, but anyway, we had aluminum versions of this as well that were anodized, and they had blue and gold and whatever. And we gave up on that idea when we ended up with blue and gold liposomes coming out the other end. This was the product development that we went through in the lab. So this is us back in the back in those days. This is Marcel down here. You can see the entrepreneurial instinct certainly started very early on. But this is, I'm going to make this point at the end, but this team, we're all still here in Vancouver. We all still work together in one way or another, and it's totally essential to have these very long-term relationships with people that you can rely on. At least they certainly rely on them to have an excellent party if you want. Now, we also knew that companies had to contribute to culture. We were concerned about that, and so we established the ballet. This is the Royal Lipex Ballet, and so the parties were really. This was a bottle of champagne in your hand as you came in the door. That's one of the nice things about having a company, is that it gives you this opportunity. $5,000 or $10,000. We were charging about $3,000 or $4,000 for these extruders, and orders kept on coming in. What were we to do? So we said, okay, well, we spent some of it on excellent. We usually use German champagne. There was a reaction. Anyway, it was a bottle of very good champagne as you came in the door, and then all hell would break loose. And so certainly the high point was the Royal Apex Ballet, and you can again see here we're doing Swine Lake. That's where we're wearing pig noses. So this was. We're still in demand. What can I say? Okay. This Lipex, which made these extruders, was bought or acquired by Northern Lipids, which is another company we established in '92. And then one day in 2015 or 2014, Evonik Industries, which is a large German company, came to the door and said, "Hey, we want to buy you." So that was by this time it was a CRO and had evolved to the point of about 50 or so employees, with a lot of interactions, biotech, pharma, etc. And so this was obviously a nice end. And anybody that thinks starting up a company is going to be a quick way to make any money, if that's one of your aims, well, this should give you some pause, because we did start this in 1985, and it was 2015 by the time that. So 30 years, this is a reasonable thing. But this is still, from my point of view, from sheer enjoyment, the best company we ever started. This is the bad, but it did turn out well. Cutis Therapeutics. So we started a Canadian liposome company. This was a company that was a subsidiary of a US-based company in Princeton. In 1987, this was to make liposomal vincristine, doxorubicin, etc. And that morphed after we all got fired. As you have to get used to getting fired. They usually get a nice severance package and all of that. We have 2 or 3 people that have been fired 3 or 4 times. As long as this came to be a mark of honor from one of the companies that we started, where the CEO was not exactly compatible. Anyway, so with INEX, we had we spun off a company called Protiva to do gene therapy. In 2000, this was where we made a very bad mistake and hired the very bad CEO. Well, this is a story in itself. Could take a couple of hours, but requires drinking heavily. And so the end result was, after this company sued INEX, and in the end they ended up winning, and all their management took over. They fired all the people that we brought into the company, all the co-founders that we had there. And so, but we were working with a company in Boston called Alnylam, and so I went down there this was in 2009, saying, "Hey, they fired us all. We want to start another company. Do you mind putting in $1 million or so a year?" "Oh yeah, wow, no problem at all." So that's where we started up Acuitas Therapeutics. So actually, getting fired was a really, really good thing. So what happened here? I've gone through most of this. We licensed the gene therapy technology, submitted vincristine, which we had a problem with. But this is where we started to use this is the top. The thing doesn't work, but anyway. We applied lipid nanoparticle technology to siRNA starting about 2005. Got into this lawsuit. Protiva won, and as I mentioned, fired all the INEX employees. We formed Acuitas Therapeutics. We developed in Acuitas lipid nanoparticle technology for Alnylam on Patro. Then there was another lawsuit. That's one thing this guy that we'd hired into Protiva was good at, was lawsuits. So they sued us and Alnylam, for that matter. But we emerged from that with a couple of $1 million and the rights to use all this technology for mRNA, which turns out to be way more valuable than for siRNA. And so Acuitas is certainly we won after all. And I should say, this is another thing. I mean, you might think bad things are happening, but it's amazing what good things can happen after that. And so we now have this is really a world's leading company in terms of enabling mRNA to be used therapeutically, and has interactions or has contracts, joint ventures with approximately 10 biotech companies in the US and in Europe. So the ugly, Arbutus Pharmaceuticals, this is the kind of thing that can happen, and as I've gone through this already, of course, but when the end of this lawsuit, where we formed Acuitas as a result of everybody being fired from INEX, then Arbutus went on under the management of the individual who had fired us all. And this is where you get into this. Sometimes it can be really quite unfortunate. I've gone through these points up to now. So Protiva fired all of our INEX or the management fired all of the INEX founders, and we formed Acuitas. So then INEX has now was acquired by a U.S.-based company they put a lot of money into it, and moved to Philadelphia in 2015. But the long and the short of it is they've raised, and I think it's closer to $700 million that's gone into INEX and Arbutus over the last 27 years. The market cap now is about $100 million, and the only asset is a small molecule drug for treatment of hepatitis B that's still preclinical. And so this is a result of monumental mismanagement from where they had a prize in their hands, and it just got, from the point of view of the investors in INEX and in Arbutus, got completely squandered. So lots of lessons learned in this. First of all, you have to always aim to be the best in the world. You may not get there, but if you don't aim for it, you certainly won't get there. Your companies evolve in unexpected ways, and so you have to be ready to roll with the punches, as it were, but just keep your long-term aim in mind. Certainly establish a good team and keep it together. Make every effort you can to keep your team together, because that's all you've got, really. If you don't have a team of people you're working with, and it's one of the delights of companies, actually, is you all work together in a very concerted way. These are relationships you never forget, and which will last a lifetime. Obviously, don't hire jerks. I've been fairly good at that over the years, and Marcel has had his experiences with that as well. It's an interesting thing. I guess, do you have any more time? 3 minutes? Everybody has 3 minutes. So I'll ask you again in 3 minutes. But anyway, you do learn something from some of these CEOs. One of the best CEOs, and Marcel alluded to this, that I ever worked with, this guy could do deals like nobody else could do deals. So I asked him one time. I said, how do you structure? How do you do this? And he said, I have a number of rules. I mean, this was not stuff you're going to learn in a business school or anything else. But his rule number one was, I never talk about money up front. That's always off the table. He said, I try to establish a relationship and say, okay, structure a deal. And there's one complete, we do this, you do that, you get this, you get that. And so, and we need something else over here. So we're going to get the deal all structured. In that process, you establish a relationship. And then say, okay, well, what's this worth? And then, there's various parameters you can pull in from market analyses and so on. And then you start to, it's okay, that's what the whole deal's worth. So if I'm doing this and that. But you're building a relationship. And in the end, it's not just your deal, it's their deal too. So it can all come together in a good way. I've used it myself, and it works. You just have to make it. You have to use that philosophy, but never think your stuff is so great you can go in and ask for $1 million, because it just doesn't work that way. Okay, never give up, but more important, have fun. I mean, if you're not having fun in this, that's what it's all about, really. If you're not having a good time doing it, it's going to be some down times, but there's some totally excellent times as well. So that's what I want to leave you with. And so thank you very much.
Thanks. Very informative talks. I just wanted to ask, if you're in the business of getting a CEO, what kind of things you have to look for in the interview?
Yeah, I think rule number one is don't hire anybody from a business background. For the startups phase, you want somebody that maybe has a PhD, has a technological background, and maybe doesn't have that much experience in running a business. But they can learn that stuff. But my experience often with people that come from a business side is that they don't really appreciate what you've got, what the core fundamentals of the company is in those early stages. And so some younger individuals who say, look, I want to do this. As long as they're reasonably compatible from many other points of view. That's where my best experience has been, is hiring people that have a PhD, whatever, in say a life science or technical field, and then want to go into business. The problem with the people coming in from the business side is that they often, the grass is always greener, and they'll say, gee, we can do this, but so over here they can do it much better. There's something wrong with our stuff, and they start to question and lose faith in their own technology because they don't understand it well enough. So that's my total bias, is that go with somebody who is more technically inclined.